we have the zoomed out idea of what the seamount looks like, but never quite know until you get down there.
It's really important to have that high resolution map though.
Yes, actually, so the satellite altimetry, which is all we have until we use our multi-beam sonar on the ship, oftentimes has a very coarse, vague idea of the shape of what's happening. So this peak actually ended up being quite a bit, several hundred meters higher than what the satellite altimetry showed, and but it was pretty true to the general cone shape, although we're finding that they had these ridges kind of radiating out in a radial pattern from the peak, which were not. It just kind of looked like a vague cone.
What are the depth divisions on the survey?
On the contour lines are 10-meter contours.
10-meter.
One of the things we didn't know before we started mapping also was whether this would be a conical pointy seamount or a geo, a flat type seamount. And so we know now from the mapping that there's not a flat structure on the top. So that indicates that when this seamount was formed, there wasn't enough magmatic injection to create a giant volcano to get it above the sea surface, which it would then cause the erosion to form a geo. So that's also, even just getting the map helps us understand the underlying geological processes that formed these mountains.
You can see in the Argus view right now kind of the idea of the structure that we're going over. It's this ridge that's going up towards the peak, and this is one of the kind of features on it.
Definitely, if you're not in quad view, I encourage you to either look at satellite 2 or the quad view on nautiluslive.org. Jake, what's your strategy as you're tracking with Hercules here?
I try to maintain a delta depth. So basically the depth or the distance vertically between Hercules and Argus of about 15 meters, give or take, depending on the swell. And then I also just try and assist the Hercules pilot with anything they need, cameras, lights, that sort of thing. And I also am checking gauges and other readings like temperature and electrical sensors to make sure everything on the vehicle's good.
So it looks like we're seeing some more dead sponge structures on the seafloor. The view from Argus, which is in the top right quadrant of the quad, it's very intriguing. You see some shadows casting over the top of this feature.
Like a sharp ridge, isn't it?
Yeah, and it's always fun to imagine what those things are on the other side. Are they other rocks? are they corals?
Yeah.
We'll have to wait a few minutes to get up over the top.
All the perfect rocks are on the other side.
Of course they are.
Yeah.
When we talk about waiting to move, I don't know who I'm going to address this to. Maybe Rennie, you can take this. But what are we waiting for?
Yeah, so we have a two-body ROV system, Argus and Hercules, and Argus it's kind of like a weight that we dangle behind the ship, and then we have a much softer tether between Argus and Hercules. Yeah, so it's kind of like wherever we put Argus, Hercules now has a leash, and so Argus is not only just the eye in the sky to give us context of geological surroundings and hazards and what have you, but it also decouples the ship's heave from the ship to Hercules. So that's how we get such steady shots. The original question was
yeah, we're talking about just kind of waiting. Are we a little bit ahead of the ship at this time?
Yeah, so you can imagine if you dangle a weight in water and you move the ship, you've got quite a bit of a pendulum before that weight responds. And so now we're 2,500 meters deep. If I move the ship, we have to wait for Argus to move along with it. So we can get that a bit steadier once we kind of get all comfortable with the pacing and everything, and maybe have a nice slow, steady pace. But the downside of that is if obviously if we have a wall or hazard, we'd have to move quickly over it. And also there's less time to stop and sample without backtracking. So we're kind of like playing a little dance here and figuring out what pacing everyone likes here as we try to get our dive objectives complete while also being able to stop and look at stuff.
Are we seeing craters on the other side?
I think that's the optical illusion. I think those are boulders.
Illusion, yeah.
But it is a really cool view right now in Argus of this ridge here.
Mm.
I think Jeff is bribing us for something here. I'm not sure what it is, but...
It's not me. I don't know who. I'm just passing it on.
Okay. Oh.
Quite a structure in front of you.
All right, wow. Nice big boulder with some corals on the side.
Throw that on the roof rack.
Might not want to stand downslope of that one.
Yeah.
It looks a little unstable.
Yeah.
Looks good.
A little bit more density of some of the filter feeders at the top of this structure.
Yeah, you can see if you watch the marine snow and how it moves through the Hercules camera, you can see it's kind of coming towards us. And oftentimes a lot of sea fans will orient their, orient the colony across the flow in order to maximize the amount of surface area that they can have to capture particles. It gives you an idea too, considering how slow some of these corals grow, that these currents, these flows are probably pretty consistent over a very long period of time. Otherwise, in some cases where we have more squirrely flows and, maybe there's reversals over periods of time, you might end up with colonies that are oriented in different directions, or they might have just inconsistent growth patterns.
We saw that really long straight one earlier.
Yeah, those are interesting because those bamboo corals, the unbranched ones, they'll typically grow straight up, right? But when they reach a certain height, the flow conditions at the top of the colony are different from the flow conditions at the bottom. And so oftentimes you'll see they'll go straight up and then they'll kink 90 degrees and just start growing with the flow.
Interesting.
What's that big one in the background there, Steve? It's kind of fluffy looking. To the right of the...
Yeah, oh, I see it, yeah. Any chance to get closer to that one?
That's a cool one, yeah. Let's go.
Higher density here. Hanging out in this gully.
Argus should be coming towards you as it slowly meanders, so you should be all right.
Roger.
Quite a current. See the tether.
Yeah, the current's a stronger one today.
Yeah. Did we ever find out what the tether length was?
30 meters, I believe.
30 meters, yeah.
Somewhere around there, yeah.
You know, this might be a good place to do a water sample for eDNA if we can get like you know, a couple of meters off the bottom or however comfortable you feel, since there's a pretty good diversity here and a decent density.
Sure
So after we do a zoom here, yeah.
You want this shot first and then we take a sample?
That sounds great.
All right. I'll try to hold you as steady as possible here, Jeff. Go ahead. Actually, let's just land.
Yeah, so there's a couple different types of coral in our view here.
All right, Jeff, go ahead and push on in there a bit, and then I'm going to adjust.
Now, is this considered a high enough density for one of those coral Niskin bottles?
A bit more.
Yeah, definitely. You know, relatively speaking, you know, when you're this deep, you kind of have to take what you can get. But this is pretty dense colonies for this depth we're at. Yeah. Any more zoom on that? Oh, thanks. Yeah. So this is a colony of Chrysogorgia.
Yes.
One of the
I got it.
One of the golden corals. Now, can I give you a better idea than that? Probably not, but it's reminiscent of something that we call Chrysogorgia scintillans, you know, which is a planar or sometimes biplanar colony. So if you'll notice, some of the Chrysogorgiids have bottle brush shapes. This one, it seems to be more planar. Yeah, that's the idea I'm going with, and I'll probably get nasty emails in the morning saying, no, it's not. But
nobody will be
that's
nasty.
what I'm going with.
All right, cool.
Is it a different coral behind on the bottom left there behind it, or is it the same with open polyps?
Yeah, I suspect it's a different one. It's probably one of these Remulogorgia militaris colonies. You know, these white fans that are more abundant.
Are you okay with a DVL reset real quick?
Yeah, sure, go ahead. Leonardo, I didn't.
So Steve, you recommended that we come a little bit off bottom to take a Niskin?
Yeah, I think, you know, maybe a meter or two off the seafloor. We want to be around, we want to be surrounded by the corals for the eDNA, so we make sure we get some of that cellular material in the water.
Sure thing.
Yeah, but we don't want to get sediment too, which is important from the thrusters. You know, we don't want to wash up any sediment into the water sample.
So I'm about 4 meters. Where I was perched there is about 2 meters altitude, but
I think this is pretty good.
Okay.
Seems like we're kind of down current too.
Did you already do a reset there, Rennie?
I did, yeah, you're good to go.
Awesome, thank you.
For those watching at home, what we're going to be doing right now is taking a water sample to for what's called a eDNA survey or an environmental DNA. And the purpose of these surveys is to show if water samples can be used as a way to identify the organisms in the environment as a simpler way to understand diversity of ecosystems. And so in an area like this, where we see many different species in a relatively high density, it's a good target to get a sample so that can then be calibrated versus what we're seeing. Because understanding, again, coral diversity in this ecosystem is an important question. Even though, again, we're not technically in the monument right now, it's good to understand what the diversity is both within the monument and just outside the monument. Because coral is an important part of the ecosystem here in the deep ocean. And corals also have a very important cultural connection for the communities that consider this area sacred. So Kalamahe, would you like to speak any more about that?
Yeah, mahalo. And so generally and stuff like that, the concept of genealogy is really important to in Hawaiian thinking and stuff like that.
What's
and so specifically, there's a cosmogony, or basically an origin chant and stuff like that. But it's actually a genealogy for one of the older chiefs in history. But it'll generally start off from, it'll basically talk about the, like from the time of darkness, or from the basically the beginning of time, and then it'll list, and then it begins to list all of the organisms that are born in sequence from like the oldest, and then it ends up all the way down 2,000 lines later at basically human beings, and then other prominent, chiefly genealogies of Hawaii. But interestingly enough, the very first thing that is mentioned in this chant, the very first organism, is the ukuko'ako'a, or the ko'a, and that's the coral polyp. And so in like Hawaiian thinking, actually we are familiarly, or we're genealogically connected to the ko'a, but also all other living things as well. And I think that's a very, and then I would also like to hear if anybody out there has other indigenous perspectives on our relationship to the biocultural resources and these environments. Please send us some comments and stuff like that. But just at the very basic level too, yeah, we descend, and we are very much so familial relatives and descendants of these things.
Yeah. Thank you for that perspective.
Oh, thank you for the question.
I think it's so amazing that we can get all that information from a water sample, and that it's a non-invasive way to characterize this region.
Yeah, the concept of eDNA is a very hot topic in conservation research as a way to understand the diversity in ecosystems. So that's why a lot of the eDNA work is funded by different parts of NOAA to see if this can be used as a management tool and a conservation tool. All right, Steve, we're going to need your expertise here again. What are we looking at over there?
I think we've got one of those upright corals or sponges we've been seeing.
Okay.
Maybe one that's a little bit more lively.
Yeah.
Is that a bathypathes next to it, right there at the lasers, or what is that?
Oh, yeah.
Oh, yeah.
I mean, the lasers, yeah.
No,
Maybe
it's not
take a
that.
look at both.
Something else, but it does look like maybe a black coral. Steve knows I like to play coral bingo. This is my Olympics when Steve is in the background.
Bathypathes is a very strong one to have on your
You're
coral
going to do a
bingo
partial
card.
zoom there, Jeff?
Yeah.
I didn't know I should bring a bingo card to this next time.
We'll have to print some out. This doesn't look like that. This is that
Oh, no.
spirally
Yeah,
one.
this is a Iridogorgia. It's another golden coral. Iridogorgia.
That spiral.
Yeah, there's a couple of different species of this genus in the area. And one of the ways we can tell morphologically between species, or we suppose we can tell between species, is how tight the helix is, how tight the coils are. Some have very long stretched out coils, and others tend to be more tightly wound. Yeah, so I would hazard a guess and say that this upright sponge on the center right is probably quite similar to a lot of these dead ones we've been seeing, like the one on the left-hand side. But the one on the center right doesn't look all that healthy either. Typically, these glass sponges will be kind of more clear, translucent, pearlescent when in life, and then they get fouled by a lot of things like sediment and other organisms as they die and decay.
Go ahead and come wide there, please, Jeff.
So there's a potential that could be alive, but I would say there are probably better candidates that we've seen on this dive so far for that show evidence of life.
And there's a very long whip structure off to our right here, and even maybe a sponge, stalked sponge over there. Oh yeah, there's all kinds of things.
Number of different organisms here.
Very interesting mix.
Surprising that a sponge can get what it needs tucked away in there like that.
Steve, what do you envision that this bigger white piece is here?
I think it's also a sponge.
Yeah.
Yeah, it just might not be of the stalked variety. Actually, we've got a couple of different cool things here. Yeah, so we've got the sponge on the right we're zooming in on now that looks to be potentially a farreid sponge, but that's just my guess from this far away.
Got any more zoom in there, Jeff?
It could be something else entirely. Sometimes we need to see the base of sponges to identify how it's attached to the substrate.
Mm-hmm.
But you know, it's sometimes texture alone will get you there. But you can see how that sponge looks much more vibrant, has a much more vibrant white color than some of the dead sponges we've been seeing. So that's a good candidate for being, you know, very much alive. Over on the left-hand side, out of this frame, is another type of glass sponge that was stalked. It looked like Caulophacus, just coming into the frame now, right there in the lower left. Yeah, that one looks like a genus called Caulophacus.
I'm gonna go ahead and come wide there, please.
And then, of course, we've got that big, long bamboo coral in the background. That's what I was talking about before, how they tend to grow up. And then once they hit that flow, it's 90 degrees. And then they'll grow like that, and sometimes for several meters.
Does that indicate kind of the optimal altitude for nutrients?
Yeah, it's tough to say. I mean, it's definitely a good altitude for them, I suppose, to feed in the strongest currents. But you also, you want to find the optimum. If you grow too high, your mass can be your downfall if they become too heavy. Yeah, another glass sponge there.
Those lasers are showing us 10 centimeters.
Yeah, the distance between those green dots, exactly.
All right, go ahead and come wide there, please.
Great. We've got good imagery of all of these. Taking pictures as I go here.
It's hard to estimate how long that bamboo coral is, but it's got to be more than a meter.
A cool angle.
You gonna do a partial in here, zoom, Jeff?
Yeah, you can see that there's a bit of directionality to how this thing has grown over time. It grows up off the rock, and then sometime in its life history, it starts to zag off to the left, grows up again, zags off in the down current direction.
All right, full wide, please.
But do you have any estimate of the growth rate of this species?
It's possible. Yeah, to do that with bamboo corals, you really need to get material close to the base, since those where the oldest material is. And in order to get that material from the base, you have to destructively sample the whole colony in most cases.
Yeah.
Well, it looks like maybe a similar species of bamboo coral here, just a few meters away.
Yep.
Very similar shape. Up and over in the current.
Yeah, this in the Pacific, these types of bamboo corals are ones that give a lot of taxonomist headaches, because bamboo corals tend to have very unusual branching patterns sometimes. And one of the things that we wish we could do more of is understand how bamboo corals grow, because they tend to You know, we tend to see them in snapshots. You know, we see this particular colony as unbranched, right? But there's really no way of knowing if this is actually an unbranched colony, you know, if it was to live to its oldest age, because some bamboo corals might not branch until they're several meters in length. And then all of a sudden you got a branch, you know, way out on the end of the colony, and it's just, it's puzzling. So that happens sometimes, and definitely some cause for concern.
We're closing in on that 2,500 meter mark. Slowly progressing our way up.
Is that depth significant?
Not necessarily, just a target. Give ourselves a reference point for how far we've made it. So since the start of the dive, we've ascended about 200 meters. And I'm not sure what distance we've traveled.
Since the start of the dive, about 460 meters, 460, and remaining, as the fish swims, would be 900 meters.
Okay, thank you. Do we need to put in another ship's move, or are we still looking pretty good?
I just called one in.
Okay, great.
Yeah, stealthily in the background.
It seems like, and I'm basing this on the detritus I'm seeing flowing across the screen, it seems like there's some significant current here. Is there any way, or do we have a current meter on the ROVs, or is there any way to measure that? Maybe I'll throw that to Jess.
Yeah, we primarily just do it visually. I don't think there is any current meter in like the ZTDs or anything like this, so I don't believe so. But visually, we can kind of do some particle tracking going across the screen, you know.
We've done that with bubbles before and methane seeps. They've had a big card that we hold out behind a plume, so you can get the you know, with deviations in the placard. And yeah, I suppose you could do the same with particles going by.
Yeah. Actually, we can just take our hands off the joysticks for a second, see how far we drift.
So
this is without any thrust.
Take a look in the Argus view. That's in the upper right quad view. You can see without any thrust or action, the direction and the speed of the current a little bit.
Maybe why it'd be awesome to be a passive swimmer.
Yeah, we'll just follow you.
Steve, you were mentioning earlier about how the polyps of the coral are filtering marine snow out of the water. Do they have any selectivity in what they want to ingest? Do they search those particles for something?
Funny you should ask that, because we've tried to experimentally observe how deep water corals feed, and it hasn't been very successful.
Okay.
And recently we tried to, so we created, along with the engineers this past summer on the ROV Falcor, where we're surveying corals in the US Phoenix Islands. We devised a piece of equipment that basically allowed us to use a syringe to push bits of food, which is basically ground up fish and squid,
Could you zoom in on those white features?
Sure.
Into a coral colony and try and see what would happen if we gave it particles, because typically it won't respond to the marine snow. This is it's a slow feeding process, but if it was given a very large, you know, nutritious chunk of food, what would it do? And
all right, Jeff, go ahead and push out.
It's very tough to get them to respond in time scales that we're down here for.
Yeah.
Yeah. Because oftentimes if you send some food their way, they're more likely to close up in a defensive posture than to, you know, start, you know, manipulating food. particles.
Speaking of a defensive posture, it looks like we might have some attached friends on these corals.
Sure, if you want to push on in there a bit more, Jeff.
Yeah, are these little squat lobsters?
They're brittle stars.
Yeah,
they're
they look like brittle stars.
Sorry.
Yeah.
Yeah, this looks like
Is that a barnacle down below?
Kind of looks like a barnacle.
You
You
can
back
see
in
the
center.
cilia.
These look like a
like that.
few species of primnoid octocorals.
Sorry, this is the most stable shot for you guys here. Come a little wide there, please,
Yeah, and
Jeff.
there's some barnacles
Thanks.
as well.
Some kind of necked barnacle.
It seems like these brittle stars are not harmful to the corals. You can see that some of the arm tips of some of these brittle stars are actually up off the coral colony.
Yeah.
And also using the coral to get up off the benthic boundary layer and get up into the flow where they can potentially capture particles and feed that way. So they're kind of, there's one hypothesis that says that the corals don't really receive a lot of benefit, but in some cases, the physical action of the brittle star moving across the colony may help dislodge things that are potentially harmful to the coral and help clean it, basically.
See that linear feature in Argus sonar?
Yeah.
Curious about that.
40 meters?
Yeah, 20, 40, yeah, just about. What's that? Headed straight for it.
Interesting feature we're coming up on here. Not really quite sure what I'm looking at yet.
Yeah, we were just noting in Argus's sonar, which is on the left here, the scanning sonar forward. See that linear feature?
Yeah.
Just 40, 45 meters out in front. And curious about that.
This almost, it's hard to really tell. It almost looks like a dike feature.
Yeah.
In the basalts.
Yeah, I see what you mean.
That's got some alteration on the side of it.
Huh.
What was the word you just said? Dike?
A dike, yeah. In these mafic settings, you can have eruptive flows where you have pillow basalts and things created, but you can also have kind of magmatic injection from within. And as that cools, it forms a feature like this called a dike. And hopefully I explained that to the extent of my geological knowledge. If there's any geologists who are listening in, you can clarify my intent there.
You love rocks, but what is your particular specialty?
Depends on the day. I have a lot of different titles: marine microbial biogeochemist. So that means I am interested in microbial life in the marine environment, and I look at it from a perspective of the interaction of biology and chemistry. And often that chemistry is derived from geological materials, hence the biogeochemistry. So I know just enough chemistry, geology, and biology to be dangerous, but I'm not an expert in any one of those fields.
For those of you listening along, that is Dr. Beth Orcutt. Check out her bio. Learn how you too can become a, oh goodness, marine biogeochemist.
Yes, there you go. Oh, looks like we have a fish above Argus. Oh, it's already out of the frame. Or not above Argus, but between Argus and Herc.
I see it, yeah. Just going over the
Rennie, I see you pointing at that feature. I'm wondering if that's like another overhang that might be above us.
We haven't seen a lot of fish potentially because of the light factor. Would that be startling to fish down here?
There are definitely fish that will want to avoid light. The lower density also could just be we're at a relatively deep depth. There's not a lot of food around here. So it's not a super yummy place to live. But surprisingly, in similar types of environments, if you were to put down a bait ball, you will see so many animals. So clearly they are often in the vicinity and have an acute sense of knowing when there's food available.
Probably any vertebrates living at this depth, they're going to have a very slow metabolism.
All right, so we're continuing to climb up this ridge feature. Definitely some big boulders. Maybe some kind of linear feature ahead of us. We'll see what it ends up being. Maybe have another bamboo coral off to the right there. Well, it looks like we have a
We have a question for the whole control van, but maybe we'll start with Beth in our science row here, but wondering what would be your personal holy grail of a find during one of these dives?
ooh, I don't know. Well, I am bringing to this exploration an assumption that these seamounts don't have any active hydrothermal circulation in them. Unlike other seamounts that I've been privileged enough to explore in other environments where seamounts can be conduits for fluid flow, both in and out of the seafloor. We know this if you've seen science documentaries with hydrothermal vents, they're really dynamic places. There's other places where water comes out at much lower temperatures. And often an indicator for that is a bunch of octopus. I'm coming to the seamount with an assumption that we're not going to find that, but that would be amazing if we did. Prove me wrong. So I guess that would be my holy grail for this dive, is if we were to come across a bunch of octopus indicating some fluid flow, but I think it's unlikely. I don't know. Steve, do you have a?
That was going to be mine as well. Like if we actually found any active venting, or even just like warm seeps, that would be kind of like, that would be a big deal.
Another type of expression of low temperature flow that's been found at the base of the underwater volcano to the southeast of Hawaii, the Big Island, which was recently given its former name. Oh, and I'm going to mess it up. I'll have to look it up. I apologize. I can't remember the name for it now. It used to be called Loihi Seamount. At the very base of that seamount, at 5,000 meters water depth, there have been discoveries of low temperature fluid flow. Very, very subtle. The temperature difference is maybe 0.2 degrees Celsius warmer than the surrounding bottom seawater, but it's enough to support a really lush environment of iron oxidizing bacteria. And if you were looking at it, it would look very similar to this. It looks like a pillow basalt, but then when you poke at it with a stick, it's soft. Very, very soft for quite a ways. I think you could stick a stick into it about 1 meter. And that was totally unexpected. The scientists who were part of that dive had gone down to the bottom to try to just understand, let's get a sample of rock from the bottom of the seamount to compare to the top. And they were not expecting to find that evidence of low temperature fluid flow. So these underwater monokai can always surprise us.
Yeah, I think, you know, we still have more mapping to do while we're out here over the next few weeks, but I think what would be striking for me is a combination of things. It would be finding a seamount that has a summit depth that is within a few hundred meters of the surface. You know, potentially what we call a mesophotic depth seamount with a summit 100 to 200 meters, something like that. That would be really interesting because there's not many of those, and I know these are much deeper seamounts in this part of the world, so I'm not optimistic that would happen. But you know, if we were to find something like that, that would be a pretty big deal because that expands the amount of the depth range that life can exist on these seamounts.
So for those watching in the quad view, if you look at the view from Argus down to Hercules, it looks like we're coming up on a very steep wall here, which maybe was whatever that sonar reflector was.
We've been using the term seamount. I don't know that we've defined exactly what we mean in our context. What qualifies and what does not qualify as a seamount in this vicinity here?
Oh, that's a fun question. I think there is a technical discrimination between a seamount and an outcrop. I don't remember what those technical differences are. So essentially what I think of it is as an underwater mountain that is surrounded by, at the base, sediment. That sediment is kind of a blanket around the bottom of the mountain that limits the flow of water in and out of the seafloor. And so these features, when they were closer to the hotspot that they formed over, might have had bare ground around them that's been slowly covered with sediment.
This is like a massive sheet flow pattern we're seeing here. It looks like you can see kind of the waves in it, and then it's been filled over with some of the rubble.
Yeah, you're right, Rennie. It does kind of, I think that's who said that. It does have that look, a little bit of a sheet flow look.
And I believe a seamount has to be base to peak at least 1,000 meters to be considered one.
Okay. Yeah, and so maybe an outcrop is below that.
There's another classification, yeah, I've seen in the literature called abyssal hills, which is 500 meters above the surrounding seafloor. But yeah, it's 1,000 meters above the surrounding seafloor is generally the cutoff that I see most commonly written about. And then there's an upper limit to these things too. Chris was mentioning this the other day, that if these seamounts come up to within a couple hundred meters of the surface, sometimes they're referred to as banks, or if they are within 5-10 meters of the surface, you know, sometimes geologically they've been just called reefs, you know, shallow potential hazards to navigation. Yeah, and then of course you have the different classifications of seamounts depending on what type of geomorphology they have, if it's a guyot or a peaked seamount.
Guyot being that flat top structure.
Yeah.
Trying to remember the terms I learned for different types of lava. So there's and pohoho. Is that correct? Anybody remember?
I know and ahoy hoy.
Ahoy hoy, thank you.
This is really cool sheet here. Kind of in the context of the mapping behind it, and we see this ridge, this loby ridge. It's cool to think about how that occurred.
It's a little something swimming in Hercules view.
Oh, yeah.
Like a snailfish or something.
Sure thing, thank you.
All right, so we're now above that 2,500 meter water depth mark. Somewhat arbitrary, but good signpost for our progress. Very interesting flow features in front of us, and it looks like it gets a little bit more rubbly just above us, based on the view and from Argus. I'm wondering if it might be worth trying to collect one of these angular samples, if we can, not necessarily in the rubble, but if we see one that
Yeah, just
like
called
might
in to
be
move,
situ.
so at some point during it, when we feel, if we see anything, I think we'll have the leash be all right.
Yeah, sure thing. What do you, for angular samples, what you looking for?
Yeah, this is more just I'm talking out loud. I don't have a good target to point you at.
Roger.
I'm just thinking that this is a different type of rocky environment. Might be a good idea to get a sample, but a lot of what we're looking at right now, it looks like rubble from somewhere else. So,
Roger.
Yeah, it looks like.
and we'll have the real geologists coming on in the next watch, so they can also make those decisions a little better.
Internet geology is not real geology.
Yes, if you're not familiar, we work in 4-hour watches, and this entire control van will change over at 12 o'clock Hawaii Standard Time. That allows the ROVs to stay underwater for around the clock, since they don't have to rest, eat, or use the restroom. But we humans will swap out. Our counterparts will be joining at 12. That'll be the 12 to 4 watch.
I'm wondering if this rebel field is not a great place for life, and we're not seeing much coral or sponge action here.
Yeah, there could be a couple of things. You know, we could be kind of in a channel that doesn't get a lot of good flow. or if these rubble pieces are loose, they have a tendency to not support corals, that grow very large at all, or it's probably a combination of factors. Obviously something that's slow growing like a coral or a sponge is not going to live very long if the substrate's not stable to support it. So if you remember back to the boulder where we took the eDNA sample, that thing wasn't going anywhere anytime soon. And so those things tend to accumulate and aggregate on those rocky surfaces that are stable. But it's very deceptive with these types of environments because there could be a lot of manganese crust in here that, is kind of forms a concrete of sorts that glues the rocks together. So it could actually be quite cohesive.
The view in Argus's sonar looks like a kaleidoscope.
Yeah, we got something that's interfering with that.
Some of the other acoustic instruments on board. Artifacts.
Wondering if it's that USBL. I can test that.
Another steep little embankment here.
I'd assume if it was USBL, it would be affecting them both.
Was that a question?
Oh, sorry. No, that's should have gone off SPL for this part. Yep. All good.
There's a stalked crinoid on the big boulder to the left. I don't know if anyone sees that. Very fine.
This right here?
Up in the middle of that rock.
Here?
Yeah.
Okay.
Yep, there's a stalked crinoid. Yellow.
Oh, yeah.
Lily.
Oh, wow.
Wow. Good eye.
Good eye there, Steve. You want to do a partial there?
These are can be quite common. These are, like I mentioned, stalked crinoids, or sea lilies. Sometimes they're called. Relatives of sea stars and sea cucumbers, sea urchins, all belong to the same group.
Go ahead and come wide there, please.
Do they have tube feet like other echinoderms?
Yep.
Interesting textures in those rocks.
Would you like to zoom on any of these rocks for texture?
Yeah, maybe these in this area over here.
Sure thing.
Beth, I'm sure you were part of the selection of this route, but were there any particular considerations taken on what ridge or why this particular route up to our end point?
There was some reasoning to our madness. In viewing the multi-beam data that was generated earlier today, this ridge seemed to have what we call scalloping, which indicates that there have been some wasting events off of the side of the mountain. And so that is usually a good indication of places where we can find fresh rocks that haven't been too highly altered, as well as places where there's a lot of current potentially for finding animals. And so that's why we pick this region.
Beth, are these bubbly rocks the one you want to see?
Yeah, they're very beautiful. But it doesn't look like there's necessarily something that we can pick up or that would be good for Andrea here. So we can go ahead and zoom out.
Okay, sure thing. Thanks, Jeff.
Just while we're waiting here for Argus is still moving a little bit more. If you take a look at HiPAC screen back there, Beth.
Yep.
I just want to know if you'd prefer to go kind of up here to this plateau, or if we can kind of, if you'd like to stay in these kind of more steeper contours over here, and we'll strafe along. And
yeah, good question. Probably more towards the steeper contours.
Sure. So I'm going, I can just kind of keep on my current track like that. That'll keep us in.
Yeah, I think that's good.
And then, yeah, okay. Another one.
Roger. If you're just tuning in, we are, I don't know, maybe approaching halfway of our intended first dive of this expedition here just outside Papahanaumokuakea Marine National Monument. And this dive we descended on an unexplored seamount. We dropped about 2,700 meters and are approaching 2,480 meters. We've been collecting rock samples for geologic and geochemical analysis, and we've been doing our best to characterize some of the biological forms that we're seeing, including coral, sponge, some invertebrates. We're kind of following a ridge. It might be hard to see from Argus or from Hercules, maybe a little bit better from Argus, but moving our way up this seamount.
It looks like there's another dead sponge feature on the seafloor to the right side of the Hercules view.
So would we think that there's significant manganese crusting on these? Or that's kind of unknown, it's just that they're black rocks until you get them up?
Yeah, some of the ones that, like when we were zoomed in, they look like they could have some crust features on them, but they look like they were pretty solidly attached to the seafloor.
Yeah, that's Andrea's. That's a tough job for Andrea because she needs the rocks that usually are hard to get.
Yep, that is true.
They're firmly attached, not altered.
Okay, there's the lasers.